the other talks that are here. And I actually made this very simple and straightforward so that I can encourage you to ask me questions afterwards, because this will be a new idea for a lot of you. Not all of you, of course. And that is the idea of using human pluripotent stem cells to treat lots of diseases, including neurological diseases. Yes, thank you. These are my disclosures. I'm founder and stockholder in Aspen Neuroscience, company I started in 2018, which is working towards an autologous dopamine neuron replacement therapy for Parkinson's disease. I'm also a stockholder in a number of companies. They all have one thing in common. They have something to do with stem cells. And I'm also on ethics advisory board for Merck, Germany. So pluripotent stem cells are unique. So those of you who haven't had the pleasure of culturing them, they are really quite remarkable once you get the hang of it, because they have some of the cancer cells. They can proliferate forever. They're essentially immortal. They're the only normal cells that are immortal. They have normal genomes, at least when they start, and they will divide forever. So it makes them a unique resource for a lot of applications. The other thing about them that is remarkable and useful is that they can become every cell type in the body. So they grow forever and they make every cell type in the body, which I found those two things very attractive for applications in human disease. There are two sources of pluripotent stem cells. Original source, and both mouse and human pluripotent stem cells was embryos. In this case, in humans, the embryo stage that's used is called a blastocyst, and it is about 5 days after fertilization and before the embryo implants into the uterus. So these embryos are commonly used at this stage for in vitro therapy. This is the stage at which they get transferred. And that's why they were available, because many more are made than are necessary for people's reproductive needs. So these were taken and cultured, and essentially this tiny little bit of cells called the inner cell mass could be coaxed into dividing forever in a culture dish and making a long-term culture of pluripotent stem cells. So that was those were first made in 1998. It took a little while, and they were first funded by the US government in 2001, which was when I launched my career in human pluripotent stem cells, right at the beginning. In 2007, something even more remarkable occurred, and that was from Shinya Yamanaka in Japan, in Kyoto. He developed a technique to take cells like dermal fibroblasts or blood cells, and by adding 4 transcription factors, essentially turning back the clock in those cells until they are exactly like embryonic stem cells. So this also was good because it avoided a lot of the controversy of using embryos, and also because you can make these cells from any person. These cells only exist in culture. Okay, these are not cells that you can get out of people's bodies. You had them once when you were 5 days old, but you've never had them since. So there are a lot of uses over the last 20 years that have been developed for these cells, and I think these are just some of the highlights. These are different cell types that you can make from iPS cells, and these are the projects in which there is actual clinical trial or close to clinical trials using these cells. Retinal pigment epithelium is already in clinical trials for treating age-related macular degeneration at the NIH. There are a lot of projects, including mine, on neurons, using neurons and glia. And these include treatment of Parkinson's disease, epilepsy, and leukodystrophies. Parkinson's disease and epilepsy are already being studied in the clinic with different cell types. My company, Blue Rock, Neurona, and Novoglia, which is the microglial company that I advise. Epidermal cells are being developed here in San Diego by a company named Stemson for hair loss. They want to build hair follicles for transplant. In the CAR T field, pluripotent stem cells are making inroads because you can make natural killer cells or T cells out of them. And those can be made into CARs. CARs can be added so that they target particular cell types. And there are a couple of companies here in San Diego, one called Fate Therapeutics and the other one Shoreline, that are developing both these cell types, mostly NK cells, for treatment of cancer. Fate has actually done clinical trials using these cells. There's another cell type called mesenchymal stem cells, which you can make from pluripotent stem cells. You may have heard them because they are also in humans, in human adults, in bone marrow and in adipose tissue and lots of organs. These are fibroblasts that sort of grow in the connective tissue between organs, between cells. But you can also make them from pluripotent stem cells, and there's some advantages to that. And they are in clinical trials, not in the US, in Canada and in Europe, for graft versus host disease. They're added along with the graft in order to suppress inflammation. There's a company called Cynata that's doing that. It's an Australian company. Now, cardiac cells have been really challenging. Several major companies have tried to, you'll find this out in a few minutes why it's so hard. Cardiac cells are, of course, for heart disorders. And companies like Blue Rock and a relatively new company called HeartWorks. HeartWorks has a permission to do a clinical trial. They have an approved IND. And then Sana is a company in the Bay Area which has dropped their program because in favor of other cancer-oriented programs. Pancreatic beta cells have burst into the consciousness, I think, of people who are interested in type 1 diabetes, because you can actually make these from pluripotent stem cells and they function just like islets. You can transplant them into the hepatic portal vein where they lodge in the capillaries of the liver. And if they survive, they will just keep making insulin, and people have been known to no longer require insulin. The main player in this field is Vertex, which has acquired a couple of companies, a couple of biotech companies that started out with this technology. So let's turn to Parkinson's disease. Parkinson's disease is caused by the loss of dopamine neurons in the part of the brain called the substantia nigra, which you can see in this diagram of the brain. The neurons from in that area project through the brain and make synapses, make contact in a part of the brain called the striatum, specifically a subpart of that called the putamen. But anyway, that's what signals those and controls those fine motor movements that go wrong in Parkinson's disease. So by the time somebody has been diagnosed with Parkinson's disease, half of those neurons are already dead. So I'm not going to talk about what kills them. They're very, very energetic cells. They require an awful lot of energy. There are lots of reasons why they are particularly susceptible to outside agents and perhaps certain characteristics like inflammation in the brain. There is a drug therapy that has been around since the 60s that increases the production of dopamine from the cells that are still left. And this improves the effectiveness of those neurons and can alleviate the symptoms, at least for a while. But there is no therapy that prevents the continued degeneration in Parkinson's disease. So there's no therapy that can stop the disease, only treat it. The reason it's so difficult to treat is really because the cells continue to die. And even when somebody is diagnosed, the best you could possibly imagine is you can save half or maybe 10% or something of their dopamine neurons, which is really not enough for them to lead a normal life. There are no drugs that specifically rescue dopamine neurons. And of course, the blood-brain barrier is always a challenge. It's difficult to get drugs across. Delivery of neurotrophic factors has actually been tried through an implanted catheter. And it helps the survival of the remaining neurons, but it had some serious side effects, including having an indwelling catheter in your brain. There was a lot of problems with infections or other brain damage. Gene therapy is also being examined for increasing the effectiveness of the remaining neurons, or even turning other cells into dopamine neurons. The problem with all gene therapies, especially in the brain, is that the delivery doesn't work very well. And there are other problems too, but starting with delivery. There has been an attempt to use an antibody therapy to remove aggregates of alpha-synuclein protein. And similar to most of the efforts to get rid of amyloid protein in Alzheimer's disease, it hasn't worked. So Parkinson's disease is a good candidate for cell replacement therapy because of its history. In the 1980s, 1990s, and 2000s, there were more than 300 people with Parkinson's disease that were treated with fetal tissue. From fetuses, these are aborted fetuses from 6 to 10 weeks old. And there's a little picture of one. You can see that it's just like this sort of curved structure. That's the brain part. And if you take out the tiny little bit of the brain that where the precursors for the dopamine neurons in the substantia nigra are, they will eventually become those cells, and put those directly into the striatum, the target of the substantia nigra neurons, you can get recovery sometimes. They act like little substantia nigra, but now they're they're straight into the target. And they will release dopamine and they can alleviate symptoms. The trouble with this was that there was no way to do any decent quality control. There were very few successes. But we were, and I think everybody in this field was buoyed by the fact that there were some successes and there were people who never had to take L-DOPA again. They were essentially cured. So a lot of groups, as I just said, there are 4 main groups who decided maybe 10 to 12 years ago to develop dopamine neurons from pluripotent stem cells and use those in therapy. And this is just a summary from a paper that was published, I believe, in 2017. This was a conference. This is very unusual because at that time we were all academic groups. This conference is a little more difficult now that that our projects are in companies. But there was a European group, Roger Barker and Malin Parmar in Sweden and the UK, a group in New York, Lorenz Studer at Sloan Kettering, and a group in Japan run by Jun Takahashi in Kyoto. And then there was the group that I was running in La Jolla at Scripps Research. And we all had the same goals. We all wanted to make cells, pluripotent stem cells, into dopamine neurons in the brain to do a much better job than using fetal tissue. You'll notice that these are highlighted differently because there's a very distinct difference between one group of these and the project that I started. All of the other ones require people to be immunosuppressed because these are not their own cells. And even though the brain was thought to be a immune privileged site, it really isn't. Cells are rejected in the brain. So when we started, we decided that because you can make iPS cells from any individual, we would develop an autologous therapy. So we would take cells from each individual, we would make iPS cells from them, and then dopamine neurons from that person, and avoid the issues of immunosuppression entirely. So this just summarizes the sort of the pros and cons, because neither one of these approaches is perfect. So autologous therapy doesn't require immunosuppression. And you also don't have to do this technology that people are now using to make the cells invisible to the immune system. They're making hypoimmune cells by knocking out the main triggers of rejection. So if you use allogeneic cells, immunosuppression is required. And also, since you're going to use just one batch of cells, you have to scale up one cell line to billions of cells. There's a challenge because of that. Because of growing the cells acquire mutations as you grow them. Notably, many cell line banks, human embryonic stem cells and iPS cells, have been discovered to carry p53 mutations, the dominant negative p53 mutations that are, of course, at the basis of maybe half of the cancers. So that means they are not a good idea for a transplant. So that means that if you grow up a lot of cells, you have to monitor them constantly, which is expensive. And also, interestingly, if it doesn't work the first time, you can't redose people with the same cell line that you gave them the first time, because they will have, even with immunosuppression, built up immunity to that particular cell line. If you're using autologous cells, in theory at least, there should be no rejection. You should be able to dose people over and over again without rejection. There's a lower probability of mutations because we only need to make a few million cells, which is honestly the size of a culture dish this big. And we, instead of doing a scale up, we do a scale out. And as I said, we have already shown actually that the cells are not rejected. So this is an overview of how to do an autologous therapy with pluripotent stem cells. So we start with somatic cells. We're starting with this person. We take a little bit of their skin, we transform those cells into induced pluripotent stem cells, and then we grow them up. So we expand them. And after we have expanded them, we turn them into a precursor of a dopamine neuron, because dopamine neurons, if you try to put those into brains, they die. They have to be at an earlier stage of development in order to survive. So they have to be dopamine neuron precursors, and then they're transplanted. So the tools that we put in place in order to make sure that an autologous therapy is safe is, first we use SNP genotyping with the person and the cells that we get from them. This is the same technology that 23andMe and Ancestry.com use. It absolutely identifies a person as that person. There's no mistakes. So we use that, but we also get information from SNP genotyping about structural integrity of the genome, which is really important to me, at least. And I think we would all agree that it's better to have an intact genome if you're going to put a cell in somebody's brain. We then do another study of, we also do whole genome sequencing at that stage, and then we do the same two things again after we've grown up the cells that we want to differentiate. We have independent clones. We don't need that many cells, and so they're vetted by doing whole genome sequencing to make sure they didn't change while they were in culture. And then at the very end, we do whole genome sequencing again before the cells are transplanted to make sure they didn't change while we are differentiating them to neurons. And we also do SNP genotyping to make sure that they're right cells. And then we have two different RNA-seq tests. One we call pluritest and the other we call neuritest. And this is a phenotypic screen for having exactly the right phenotype of cells using a whole genome gene expression profiling. We create a database of hundreds of samples that we know will have the characteristics we want. And we use machine learning to put in, compare other samples to that database. And they either pass or fail. So that's pretty straightforward. Okay, so there has been progress in the trials. Kyoto has transplanted perhaps as many as 4 of the 7 PD patients that they plan to use in their trial. Sloan Kettering and Blue Rock Therapeutics, which is using an embryonic stem cell line, has transplanted perhaps 2 of 10 patients. The reason those are question marks is because I only find out about the transplantation when I talk to the actual people doing the work. It's not published. And then Lund University, Sweden, has just gotten approved by the Swedish Medical Products Agency to start a phase 1 trial using an embryonic stem cell line, a different one. And then our study is still in the IND enabling safety and efficacy studies. We're moving through the FDA process, and that's all I'm going to tell you about Aspen because we're at a rather sensitive stage right now. Okay, so let's look at this autologous versus allogeneic idea a little bit more. Autologous, no rejection. It's possible you get better engraftment. And if you've studied embryology, you know that there's a lot of self recognition that happens during differentiation. So we are, we think that you will get a better regrowth of axons in an autologous combination. And that has been shown in a non-human primate model to be the case, but there's not a lot of other information. We have, whoops, my computer is doing weird things. We have other, of course, we limited expansion. The challenges is coming up with the reproducible methods and the QC for production of the cells from each person. And I've already told you that we rely on genomics. And it's not appropriate for acute disorders because it takes a while to do this. It could take up to 6 months to generate the cells for a transplant for a person. So it's not something that you can do as soon as somebody is diagnosed. Parkinson's disease, type 1 diabetes, there are a lot of diseases that are not acute. The advantages of allogeneic is, of course, is that it's off the shelf. You don't have to make it. You can rapidly treat acute disorders. The challenges are rejection, which is, I think, a very big deal for some people because of the immunosuppression, or that you need to make the cells hypoimmune. And you also have a higher chance of mutations, which makes it a lot more costly. And I don't seem to be moving anywhere from this. Oop, I'm stuck. Oh, there it is. Okay, thank you. I just tried a lot of different keys. So you need to ask, if you're thinking about a pluripotent stem cell therapy, you need to think of 3 basic questions. What is the mechanism action? What do you think it's going to, how do you think it's going to work before you start? Is the engraftment you need a transient thing like CAR T therapy, or do the cells need to stay there for the lifetime or at least many years in that person in order to work? How many cells are required? Do you need millions of cells or do you need billions of cells? Is immunosuppression or loss of immune surveillance, which is what happens when you have hypoimmune cells, is it an acceptable risk? Again. And so I'm just going through I'm going to show you the same list of therapies, but now and indicate which are which. The age-related macular degeneration, neurons, the hair loss are all, and cardiac cells and pancreatic beta cells are all durable. You want cells to remain in place for the rest of the life of that person, hopefully. But other things like natural killer cells and these mesenchymal stem cells that are used for graft versus host disease can be transient. So that helps you decide what kind of therapy you want to do. So if you want to do an allogeneic therapy, this is the kind of situation makes it, I think, not so risky. When you need billions of cells, because we can't make billions of cells from each individual, so we cannot do autologous therapies. But also, even if you're going to do a durable therapy, you still need to have a lot of cells. And that is just not practical if you're making the cells from each individual person. These durable approaches can be made autologous and are being made autologous by researchers. The retinal pigment epithelium is an autologous therapy. Our Parkinson's disease therapy is autologous, and the hair loss using epidermal cells, is also going to at least be tried autologously. So I just want to summarize by saying that cell therapies using pluripotent stem cells are now entering the clinic. They first started getting used by people in 2001, and now 20 years later, they're actually going into clinical trials. They can be used in other ways besides the ones I described. They can be used to replace cells that are killed by disease, like Parkinson's and type 1 diabetes, but you can also use them to deliver things. You can treat, for example, leukodystrophy by putting a cell type into the brain or into a periphery that makes the protein that is not being made by the patient. So they can be used to deliver proteins for genetic disorders. They also can be used for delivery of genes. It's a little more complicated, but it's not that difficult. And you can use them to deliver things. You can deliver the cells into a place where you want to kill something. So you see there are lots of opportunities in this field. Excuse me. So I've said this a whole lot of times already, so I'll just say one more time, that autologous therapy is preferred because of you don't need to immunosuppress the people. And if the transplant is to be durable. Allogeneic therapy is preferred if you have a transient mechanism required, or if you need to treat the person right away. So with that, I want to thank more people than are on this slide. My lab at Scripps, this is the core group that I took to Aspen to start the company. I've had a lot of support from a group called the Summit for Stem Cell Foundation. I've gotten more than $17 million from CERN when I was at Scripps, from the NIH, from the Silverstein Foundation. And then when I started Aspen Neuroscience, I have lots of investors, but some of them are really quite remarkable, including Arch Ventures, OrbiMed, and Sam Altman, who is the head of OpenAI. So that is a very interesting kind of investment. I have some very important collaborators in Wales and in Germany. And well, thank you.